In this facility, I provide different kind of help to researchers. Either if you're running a short experiment or a pilot experiment, I can run the experiment for you. And we can test out what method, what instrument and what method would work best for you. But if you're going to run a large set of experiments, it's better for the researchers to run their own experiments. And then I will train people and guide them as best as I can so they can use the instruments themselves.
Researchers at MultiPark have access to state-of-the-art equipment for sorting and analyzing cells, as well as for molecular screening. The shared technical platforms give opportunities to many research groups.
The cells are now in the sample chamber, and they're sucked up through the sample line into the flow cell, where the actual analysis of the cell takes place. In the flow cell, the cells are hit by the lasers, and in this case we have a blue, a yellow, green and a red laser. When the sample has been analyzed, depending on its fluorescent properties, the cells that are in this thin stream, and the stream is divided into drops and the cells enter the sort chamber. The drops can be monitored on the computer screen, and the cells in the drops can be sorted into tubes or into microtiter plates. So you can actually sort out one single cell into a tube or a microtiter plate.
The drops then get sorted, and you can monitor them in detail on the computer screen.
I'm now sitting in front of the Celomics array scan, and it's basically a fluorescence microscope where you can take images of your fluorescently labeled cells. Attached to this microscope, there's a software that can automatically analyze the fluorescence intensity of the images that you take. So if you compare this microscope to the flow cytometer, they both measure the fluorescence intensity of the cells that you analyze, but on the microscope you can also analyze the subcellular localization of the labeled target. This is an example of how we analyze neurons using this software. First, we identify the nuclei of the cells. The nuclei are here labeled with a blue DNA stain. Then we take away the nuclei that are not valid. Next, we identify the cell bodies that were green, so that we only see the cells expressing the green marker. And those cells are the neuronal cells.
The most powerful thing about the Celomics array scan is that you can scan a lot of samples in a short time and get an enormous amount of data out of them. But it's not only possible to analyze cells, you can also analyze a whole organism, like a zebrafish.
The algorithm analyzes, first it identifies the whole fish, and then it analyzes the red spots, which in this case is cancer cells that the researcher wants to monitor.
Here, the red dots show that the cancer has spread throughout the tail. With even more key infrastructure on the way, MultiPark is making sure that scientists can share the use of expensive equipment, advanced technologies that otherwise would be out of reach for most groups.